Right, I'd like to express my gratitude for the invitation to come here today. My name's John Anderson from the UCL Great Ormond Street Institute of Child Health, and I've been asked to moderate this little mini symposium or discussion on the interesting area of CAR T cells. I'm joined by my colleague Francesca del Bufalo from Bambino Gesù Hospital in Rome, who's flown in this morning. And then later on, I'll be joined by Gavin Lindberg and Alan Pearson, who are parent advocates who are going to join us for a panel discussion. And Francesca and I decided to make our talks relatively short to allow plenty of time for discussion. And I'm going to attempt in my short 20 or so slides to explain the technology in language which everyone will understand, I hope. I hope I succeed and I don't bore you. And I've put in some movies and things as well to try and make it interesting. So shall I press next? That's me. That's my title. And this is first depiction of how CAR T cells could potentially work. It was meant to be animated, so I'm a little bit worried about the movie, which is on the right-hand side, but hopefully the movie on the right-hand side will work in a minute. To get your eye in, look on the left-hand side. This is microscopy, and the yellow blobs are actually made up of individual cells grown together like in a ball of cells. And using this particular technology, when one of the yellow cells dies, it turns to a red color. But you'll also see there are some green and blue blobs, and those are CAR T cells. And the reason you have this sort of before and after picture on the left is because you have got cells which have gone from yellow to red as they've died. Now, the movie seems to have started, which I didn't want it to do, because I wanted you to look at a particular blob, which I can't point to because I haven't got a mouse, but it's this one, that one there, because when I set the animation off, you should fix your eye on that one, and you'll see that the CAR T cells next to it are sort of holding on to the tumor cells, and one of them is going to turn from yellow to red, indicating that it's died. And out of the corner of your eyes, you'll see lots of other little ones just flitting around randomly, some of them in a very meaningful way and others in a non-meaningful way. But if you try, so it worked, didn't it? Now, can the people at the back press return maybe and see? Oh, there you go, exactly. So there you are. So you've got the blue one, which the CAR T cell killing, and there it goes red. Did you see that? Excellent. So this obviously is a fried egg, but imagine that the fried egg is a cell, and so the yellow is the nucleus, the center of a cell, which contains the DNA, which makes the CAR, basically. In the middle is depiction of what a chimeric antigen receptor looks like. And so these green objects are found on the surface, which is the cell membrane of a cell. So how do they get there? So we take our cell, our fried egg, and within the yellow part, the nucleus are the chromosomes, and the molecule which is contained within the chromosomes is DNA. The function of DNA is to encode for proteins. It does other things as well, of course, but largely it encodes for proteins, shown as this sort of blob. And the particular protein which we're interested in making the cells make is the CAR, as depicted there in the middle. And those CAR molecules, which are proteins, end up on the surface of the cell. So if you think about the edge of a cell, or the edge of a fried egg, having this, what we call a membrane, on the outside is half of the CAR molecule, and the inside is the other half. And the reason we do that is because the CAR forms an interaction with what we call its target. And the immunological names we use is the antigen is the target, and the part of a CAR molecule which recognizes the antigen is actually an antibody. So we heard already about antibodies binding to GD2, anti-GD2 antibodies. This is basically what we do in a CAR T-cell technology. We take the antibody part of an anti-GD2 antibody and put it into a single protein, half of which is outside the cell, half of which is inside the cell. So the reason we do that is because if we then cause an interaction between a CAR T cell, which is the green cell shown at the bottom, apologies for the lack of pointer, and the tumour cell, which is the red cell. They form this close interaction. And within that interaction, individual CAR molecules, which you can see by the purple bits, are binding individual GD2 molecules shown as the red bits. And it's not just one of them, but as depicted there, there's several. In fact, within this interaction, exactly the same interaction we just saw on the video, there's probably about 100 or so molecules involved in it, and they happen very quickly and very rapidly, and it leads to the death of a tumour cell in a very specific way. Now, these CAR molecules have evolved over the last 10 to 15 years, with the very first ones being called first generation and subsequently the second and third generation iterations. The difference between them is the amount of what we call the functional or signaling parts within the inside of a cell, so the second and third generations. And all of these have been tested clinically, but how do we actually get this inside a patient and get it to work? So I've already said that the genes, which is the DNA, which is in the chromosomes, are the bit which encode these proteins. So if we can genetically modify the genes using different technologies, which has to be done outside the body, then we can inject the cells back into the patient in accordance with this schematic shown here. So the first part of the process would be the removal of all the blood cells from the body, or all the white blood cells from the body, in a process called apheresis or leukapheresis, which many of you will be familiar with because of the apheresis we use for stem cell collections. And then the genetic modification, which occurs on a patient-by-patient basis in a specialized laboratory, testing and growing up of these to very large numbers, and turning it from an experimental product into a medicinal product, which is approved for human use. That can then be usually frozen down and preserved prior to the administration to the patient. But prior to that administration, we typically give chemotherapy at an intermediate dose in order to create space within the blood compartment for these T cells to expand. The T cells then traffic to the tumour, and in theory, they'll cause a regression of a tumour. Now, unlike other types of medicines, we think of this as a living drug. So the cells are alive and potentially can be long-lived and memory cells, so they could potentially provide lifelong protection from the disease. Now, one of the consequences of injecting these charged up immune cells called T cells into the blood is that they recognize things as foreign and they start to make copies of themselves, which is necessary for the long-term persistence and for the actual tumor shrinkage. But as a result of that, they release a lot of chemicals. We call the chemicals cytokines, and if there's large numbers of them, you get symptoms which are essentially flu-like symptoms of very high fevers and drops in blood pressure and things like this, but also potentially occasionally causing neurological type symptoms. And so we actually classify these symptoms in terms of what we call cytokine release syndrome classification, with the most severe forms leading to a requirement to go to an intensive care unit for assistance with breathing and things like that. But there's also concerns about whether there might be other less acute but more significant longer-term effects. For example, the type of CAR T-cell treatment which has had the biggest global impact thus far is against leukemia, and one of the side effects of that is that the T cells also cross-react with healthy B cells. So patients who get the leukemia CAR T cells, they require treatments to make up for the lack of normal healthy B cells. B cells are antibody-producing cells, so patients who have successful treatment with this end up getting monthly infusions of antibodies. But there's also concerns in the leukemia studies. There's some cross-reactivity with healthy cells in the brain, and so there's actually some neurological toxicity associated with this. So there's definitely an upside and a downside to this technology as a therapeutic. So as I said, in leukemia was the area where this first took great interest. I'd been working on it in the field for about 3 or 4 years at the time when we saw the first exciting results of patients getting real clinical benefit, especially in leukemia. This was Emily Whitehead, who was one of the first children treated at Children's Hospital Philadelphia and has become something of a signature girl for the field. I had the pleasure of meeting her a couple of times. various meetings. So what about solid cancers? Can we have the same successes in solid cancers which have been seen in the leukemias? And we are fortunate in neuroblastoma, but we've known about GD2 for many years because of the experience of targeting GD2 with the antibodies. And as I said, we actually take the anti-GD2 antibody used in dinutuximab and redeploy it within the chimeric antigen receptor, genetically engineer the cells to have the same kind of specificity as the dinutuximab anti-GD2 antibody. And when we first started out working in this area, we were concerned about pain, of course, because we all know that our anti-GD2 antibody treatments can cause pain. So we would fully expected the CAR T cells to have the same problems, and therefore designed the initial trials to try and ensure there was patient safety. This is just one of the trials, the one which I happened to be involved in at our own hospital in Great Ormond Street. And what it showed at bottom left is that all these patients got nice expansion of the T cells in the blood, but they were short-lived rather than the long-lived cells which we had hoped for. And so the shrinkages, as shown at the bottom, were rather short-lived shrinkages. So proof of concept it worked, and there wasn't the pain which we feared, but there wasn't the sustained engraftment which we needed. And that was one of several, and I just show an example here of 4, but there are others which I could have put up in this slide, which have been completed, or which have shown the results showing a similar finding, but this is well tolerated, shows evidence of efficacy. But at the bottom is the trial from our colleagues in Rome, and Francesca is going to talk about that trial in more detail after I've finished my presentation, because the results are interesting and exciting. So we've been thinking for some time about how to improve the persistence of these cells and what can be done to enhance their function. And we thought that these 4 categories of the sorts of changes which could be done to make the successes in solid tumors which we've seen in leukemias. One of them, shown top right in green, is to change the actual, fantastic, thank you, to change the type of cell we use. Do they have to be T cells? Can we use other kinds of cells? There's a lot of interest in this. Friends of mine in various universities are working on this, especially in NK cells. There's engineering, so not just putting the CAR molecule in, but putting other genes or other genetic modifications which turns them into long-lived cells. That looks extremely promising and is now entering clinical evaluation. And there's giving combinations, so like most treatments, they often work better in combination, so why not give it in combination with small molecule drugs which could enhance their persistence or target the tumor. And finally, for neuroblastoma, we should think about other antigens which are also present on the cell surface, which might be good targets as well. And one of there's a lot of metaphors, as you can imagine, about CARs, and one of the popular ones is the armored car. And the idea here is that you get the CAR T cells not do their normal killing, but actually to spew out other molecules into their environment, a bit like a tank firing its gun, right? Fires rockets at all the cells around it. So that's actually a great concept and is being taken forward clinically and showing a lot of interest and excitement. And I'll talk about one of the trials which does that in a minute. So these are some of the current European clinical trials, and I've restricted it to Europe because it's probably what's of some interest to this audience. As I mentioned, Francesca will talk about the first one in a minute. Our friend Claudia Rossig in Germany has developed an anti-GD2 CAR, which has got this armoring in, one of the types of armoring, which the first patient of which was treated almost a year ago now, and they're approaching the end of their first phase of recruitment. And so the results from that will be made known in about 6 months or maybe less. There's a trial open in our own hospital, Great Ormond Street, which also involves this armoring. Again, it's an anti-GD2 CAR. And there's an interest in targeting other antigens, one of the most popular of which is B7H3, because it's expressed not only in neuroblastoma, but in most childhood solid tumors and also brain tumors. So the trial in our hospital, Great Ormond Street, is called Magneto for short. And it involves some quite significant changes in that rather than just having one virus infecting the cells, there's actually two. And the reason there's two is because there are actually 4 different proteins being expressed rather than just the CAR. So the additional ones make the CAR more long-lived and are designed to create more inflammation, the sort of cytokine release syndrome. But that trial has only been recruiting for 4 months, and only 3 patients so far have had T-cell products manufactured. So there's no data, that's the preclinical data, to show that it's significantly better than the previous Great Ormond Street trial with the same anti-GD2 antibody. And Claudia's trial in Germany, which I mentioned, has got this cytokine. So this is an example of the armored CAR, the armoring. And the cytokine for this particular one is called interleukin-18 or IL-18. And it's a clever design in as much as the T cells produce this cytokine, but only when they get into the tumor. In the tumor, they receive a signal which triggers them to make the second product. So the first product is the CAR. That gets, the T cells get into the tumor, get activated as a result of recognize the tumor, and then produce this other molecule called IL-18, which significantly enhances their persistence, but also activates the other tumor-killing cells within the tumor environment. As I said, that is now recruiting. And the other aspect of it, which is innovative and important, is decentralized manufacture. So there are several trial centers, 2 of which have a capacity for the manufacture of the T cells. And so rather than the patients having to travel all the time, with this paradigm, the patient can have their apheresis performed at their own hospital. It gets transferred to a manufacturing hospital, and then the cell product transferred back. This is the trial which will open in our own hospital, targeting the different antigen called B7H3, which will be applicable to other cancers as well as neuroblastoma. And it's, again, a modification of the same concept, but now there's a modification of the CAR itself so that it can be regulated by addition of an anti-cancer drug, which is called an IMiD drug. And this one won't open until the end of this year. So in the United States, I am aware of most of the studies which are ongoing and know most of the investigators, and there's a lot of activity which is very similar to the European activity. But all these are trials which are in the sort of phase 1 setting, and they have all been modified from what we learned in the first generation of trials to include these sorts of armoring technologies with a view to making them long-lived and to enhancing the amount of inflammation and tumor-killing properties. And just the final example of these sort of things comes from our colleague Andres Hexi in Baylor College of Medicine in Houston, Texas, who evaluated the armoring, but within the context of a different cell type. These are called NKT cells. The number of responses was only 3 out of 12, but actually they have gone on to have very significant enhancement of the cell function. So the same armoring has been put into an anti-GD2 CAR for their current study. And I think that was my final slide. So the summary is has been exponential growth in the interest of these in the last 5 years, following from the successes. All of these approaches are beginning to show great promise, especially in all the preclinical studies. To date, the toxicity has been much better than expected. And what we need now is greater focusing on the technologies and rapid clinical evaluation so that we can move forward most quickly. So I'm going to invite Francesca to come up. We have a point.
Thanks a lot.
And then we'll have the Q&A after.
Yeah, thank you. So good morning, everyone, and thanks for inviting me. It's a true honor for me to be here. And as John introduced you, I will talk about the results that we obtained with our trial and when we are going nowadays. But before starting with the results, I would like to put in context the experience that we had, starting to say that, as John just mentioned you, CAR T cells have obtained remarkable results, especially in the hematological fields, so for patients who were having especially leukemia. But when we move to the solid tumor field, as neuroblastoma is, the scenario is completely different and the challenge is much higher for CAR T cells. I will not go through all the details of this, but this picture is just to show you that the organization and the structure of a solid tumor is much more complex than what we have in leukemia, and several elements of a solid tumor are able to impair the activity of T cells, and this is the reason why CAR T cells have been giving less success in the solid tumor field than in the hematological field. And keeping this in mind, when we started to think about how to develop our CAR construct for fighting neuroblastoma, we decided that we had the feeling that we needed to create a more powerful construct than what was used for the hematological field. And to empower it, what we decided to do back then in 2013 was to create a third-generation construct. John just explained you about that. It's a construct that it's containing not only one, but two co-stimulatory molecules, as you can see here. So these two molecules give more power of activation to our CAR. And at the same time, by creating a more powerful CAR, we didn't know what kind of toxicity we would have faced using this more powerful CAR. And so, of course, we didn't want to go with an atomic bomb into the patients without having any tool to control it. And so, to improve the safety window of our approach, we decided to include a second gene into our construct, which is it's called inducible caspase 9, and it's what is called a safety switch. So it's a true switch that we can activate by giving another molecule that otherwise has no other function, which is called remidoside. And once we give this remidoside to a patient, this is uptaken by the CAR T cells and tells the CAR T cells to go into suicide. So the CAR T cells immediately die and they stop being active. And this is very powerful, a very powerful tool to control toxicity, as I will show you later. So with this construct, we opened our first trial, which was a phase 1, phase 2 trial. What does it mean? It means that when you get from the lab with the new components to treat a patient, you obviously don't know what is the right dose that you can give. And by right dose, we mean a dose that it's able to do its activity, but at the same time that is not too toxic for the patient. And to find what is the right dose to give to our patients, we normally start with the different designs of the study. We started with this design, so we decided to start with a lower dose of 3 million of CAR T cells per kg of recipient body weight, but we had the possibility of de-escalating down to 1 million in case we were observing too much toxicity, or escalating up to 10 millions in case we didn't observe toxicity that we felt were more relevant than the efficacy of the treatment. And once we completed the first phase, which is the one that I just mentioned, we got into the second phase of the study, where we treated more patients at the recommended dose that we identified in the phase 1. And as John was explaining you, all of our patients receive, previous to the infusion of CAR T cells, lymphodepleting chemotherapy, which is a combination of fludarabine and cyclophosphamide to create the best environment for CAR T cells to expand into the body. And we analyzed, once we opened and we started to treat the patients, we decided to do an interim analysis to see how to really have a feeling of exactly how our patients were going with the treatment. And so we analyzed the first 27 patients enrolled in the trial, and the results have been published last year. As you can see, amongst these 27 patients, we have patients, mostly patients that were treated with a heavily advanced pretreated disease. So the median number of previous lines of treatment is 3, with some patients that received up to 6 lines of therapy before getting to CAR T cells. Half of them were having a relapsed disease, and a quarter of the cohort was having a MECAN amplification. And what did we find in this first analysis that we did? First of all, that we could manufacture the CAR T cells for all of our patients. So in terms of feasibility, because that was one of the big questions before, was it possible to manufacture CAR T cells from patients that were anyway pretreated a lot with chemotherapies? And the answer likely is yes. We were able to produce multiple doses of CAR T cells for all of the patients that we enrolled in the trial. And I can tell you now that we have updated updated the cohort of patients. We have treated more than 50 patients, and out of all of these patients, only in one case we were not able to produce the CAR T cells. So it's a very good result for us, and it means that it's something that it's possible to push forward. And then, of course, we wanted to evaluate the toxicity of the treatment, first of all, as I was mentioning before. And what we found out is that surely the treatment has some acute toxicities, which are important. First of all of the patients develop a severe hematological toxicity. This means that the bone marrow of the patient is impaired in its activity by the inflammation induced by the CAR T cells, and so the patients have anemia, neutropenia, and thrombocytopenia that are severe and can last even longer in some of the patients. So, of course, there is a window of in which we need to keep the patients in a safer space because of this. And not only this, as John was mentioning, we observed the cytokine release syndrome, so the inflammatory syndrome that is caused by the activation of CAR T cells in the majority of our patients, although most of them had only fever and a requirement of low doses of oxygen. Only in one case, we had a patient that developed a higher requirement for oxygen and was treated with the drug that is normally used to treat CRS, which is tocilizumab, and he recovered completely. In this cohort of patients, we didn't observe any neurotoxicity, and of course, we observed peripheral nerve pain as expected with this treatment. From the analysis of the data of the first 9 patients enrolled in the phase 1 portion of the study, we didn't observe any toxicity that was beyond the limits that we put in our design. And so the recommended dose for the phase 2 and the dose that we are actually using for treating patients now is 10 million of transduced CAR T cells per kg of recipient body weight. Sorry, the design has been messed up by the change of computer, but anyway, in terms of clinical responses, we observe a complete response, so a complete clearance of the disease in 9 of the patients, whereas 8 additional patients had a partial response. So the tumor shrunk significantly, although it didn't reach a complete remission. And this means that the response rate in this cohort of patients is in the order of 63%. But of course, we know that some patients can respond and then relapse of the disease. So overall response rate is an important indicator of the activity of CAR T cells, but we need to know whether these cells can then lead to a long-term control of the disease, which is what we ultimately really look for. And we can define these by looking at these curves. These are survival curves, and the higher the curve, the higher the survival of the patients, especially when we talk about event-free survival, which is the survival without having a relapse or a recurrence of the disease. And in this cohort of patients, we observed that the probability of remaining up to 3 years without disease in this cohort of patients is in the order of 36%. That anyway was comparing favorably to the general observation of approximately 10-15% of this cohort of patients. But when we were treating our children, what we really observe is that when we're treating the patients with a high disease burden, so with a lot of sites of disease or a high infiltration of different sites of disease, the patients were responding as well, but all of them were ultimately relapsing after some times. And so when we analyzed our cohort, we wanted to understand what was the impact of the disease burden at the moment of infusion, and we analyzed our cohort evaluating the survival depending on different threshold of disease. And what we found out is, sorry, it's this one. What we found out is that when the patients were treating with what we called at the end the low disease burden, which was identified as a SIUPAN skeletal score of 7 or below, or a tumor mass with the longest diameter of 5 centimeters, below 5 centimeters, or an infiltration in the bone marrow below 50%, the survival curves that you can see now are higher than the patients that are treated with what we call a high disease burden. And this means that the moment at which we administer CAR T cells and the burden of the disease, it's really important to drive the function of the CAR T cell. And we have now updated the cohort. This is an intermediate update. We have now treated more than 50 patients, as I was mentioning before, but this is the results of the first 49. The characteristic of the cohort is basically the same. The only difference is in the disease burden, because of course, once we found out that the low disease burden was impacting on the activity, we changed a little bit how we position our CAR T-cell in the treatment. And before giving CAR T-cell, we aim to reach the low disease burden with other reduction strategies. And again, we evaluated all the manufacturing of our products. I will not bore you with all this data, but this is just to tell you that, again, the total cell number that we obtain are very high, in the order of 3 billion of cells per each patient. And out of these 3 billions, approximately 65% are represented by CAR T cells. So the numbers are very high. And again, in terms of toxicity, we observed the same profile that we have shown you in the first analysis, so mainly severe hematological toxicity and CRS of low grade. But in this updated cohort, we have now 8 patients that develop a central neurotoxicity, so a syndrome, an inflammatory syndrome that is involving the brain, and it's characterized by, in the milder cases, by headache or slower reaction to the stimuli, whereas in the patients that develop grade 3 neurotoxicity, we have 4, they actually had a quick loss of consciousness that required a stronger intervention. So in all of these patients, we needed to administer steroids, and since we were not seeing a rapid response, we administer also the Remidoside to activate the suicide gene. And this is what happens to these patients. These graphs are showing you how the CAR T cell in the peripheral blood of this patient go. And this is a picture of what is happening with the CAR T cells in the body of the patients. And as you can see, in all of them, at the moment of the peak of toxicity, we were having a peak of CAR T cells that were very high in the blood. So we started the steroids, and then we administered in all of them 1 or more doses of Remidoside. And what you can see is that immediately after administering the Remidoside, this is only 4 hours after, the CAR T cell dramatically drop in the blood. But what is more important, from the clinical point of view, is that all of these 4 children had a very quick recovery of their neurological function, and they became awake again within 12 hours after infusion. So the treatment is very effective at controlling the neurotoxicity. Of course, the backside of this therapy is that if we take off the CAR T cells, they may stop working, and this is something that we are still trying to understand. Of course, for this reason, we use it only in case of toxicities that we feel are really worrisome. But to date, we have some data that are telling us that this may not be really the end of CAR T cell activity, because in some of our patients, we have seen a later reoccurrence and re-expansion of CAR T cells, because not all of the CAR T cells are eliminated, but we keep seeing a small amount of CAR T cells. And as you can see, for example, in the first patient, they can re-expand subsequently. And more importantly, these patients continue to have a response to the treatment. So of course, we cannot say whether it's because of the CAR T cell are still working or whether they did all the work that they need to do before, but this is a good sign for us. And again, the response of this updated cohort confirms what we saw before, and so the response rate is very high in this cohort of patients. You need to take into consideration that here we have treated also 7 patients that had finished the first line without disease. So of course they are included in the complete remission at the end of treatment, but they were in complete remission also before. And in 7 patients we obtained a partial response. And the event-free survival at 5 years is now in the order of 60%. Sorry, I messed up with the 2. And but now we in updated cohort, we started to analyze also other factors, that clinical factors that may impact on the response. Of course, we have treated still few patients, so these data are very preliminary, and we will have to confirm them in larger number of patients. But we wanted to understand at what point we need to treat to give CAR T cells, and if there are some conditions that are favoring the response to CAR T cells, so that we can really optimize the treatment. And so, first of all, we evaluated whether the treatment given to refractory or to relapsed patients was giving different results, but we didn't obtain a significant difference in this case. And also, an important question that we had was whether the previous treatment with dinutuximab beta could impair the activity of CAR T cells for masking the antigen or inducing a refractoriness to the treatment, but again, we didn't see any difference. And also, in terms of MECAN amplification, we don't have a response. Whereas what we saw that could make a difference in the outcome is the timing of administration of CAR T cells. In other words, if we give CAR T cells early in the treatment, which means not after 3, 4, or 5 lines of therapies, the probability that they have to give better results is higher than if we give them later in the course of the disease. And this is probably, we are studying this data, but this is probably related to what we all in oncology know, and is the fact that the more we treat the tumor, the more the resistant cells are selected. And so what is the current situation with the GD2 CAR T project? Because you may wonder about this. So we have completed the enrollment in the trial, and the results will hopefully be published soon. But since we were having these important results, we felt that we could not stop the treatment in the window between this trial and the next trial. And so we currently are very lucky, because in Italy we have a legislation that enables us to treat patients in even outside of a clinical trial setting if there is an unmet medical need, like neuroblastoma, and if there is a treatment that has shown promise for these patients. And so we now are treating patients under hospital exemption, waiting for the next trial. The backside is that the treatment is approved only in Rome, so the patients need to travel in Rome, which is challenging. And currently, the way it works is a two-step process. So patients and their family, of course, need to come to Rome for a first evaluation, in which we evaluate whether the patients can receive the treatment, and we collect the T cells to produce the CAR. This lasts normally, we keep it as short as possible, so normally it takes 2 to 3 days. Then the patient goes back to his house and to his treating center. We agree together with the treating colleagues on a bridging treatment to control the disease during the manufacturing, because the manufacturing normally takes around 1 month. And then when the cells are ready and we receive the approval of our Italian regulatory agency, the patient travels back to Rome, where we need to screen the disease, and then it's admitted for the infusion. But where are we taking this project next? We want to further evaluate the efficacy of the treatment in a multi-center setting for 2 reasons. First of all, to confirm that what we obtained can be obtained across all the different centers, and to increase the accessibility to the treatment. And so we are planning an international multi-center clinical trial. These are the centers that express their interest, but other centers have emailed us about their interest, so this may be updated in the next future. And just an idea of which cohort of patients will be enrolled in the treatment. They will have to have, of course, a diagnosis of high-risk neuroblastoma, who relapse or progress either during or after the first-line treatment, and no more than 2 previous lines of therapy will be allowed for this trial. And the patients will have to have achieved what I explained you before, the low disease burden. 2 last very quick comments. Considering the results that we obtained for neuroblastoma, we decided to test whether this approach could be effective at curing also other children, not only children with neuroblastoma. And so we activated a trial to test the activity of this product also in brain tumors. Of course, brain tumors are a completely different world than the other solid tumors, so we have to start back from the phase 1 and to understand whether what is the right dose for these patients that don't give enough toxicity. So again, we are now in the process of evaluating what is the safe dose. And we have 3 arms that are now all open, treating basically all the histologies that can be diagnosed in the brain setting. And one very last point, in our experience, we had a few patients which relapsed, in which we had a relapse after GD2 CAR T, but the patients were doing very well, and so we decided to give them another try with another trial. And also we had a few patients in which we could not collect, that were referred mainly from abroad, in which we could not collect the lymphocytes for the manufacturing. And so for these patients, we were facing the challenge of what opportunity we could offer them. And we decided to try to create the CAR T cells using not the lymphocytes of the patient, but the lymphocytes donated from a donor. It's a very complicated process. It requires previous administration of an hematopoietic stem cell transplantation from a different donor than the patient, so it's an allogeneic transplantation. But what we found out with this approach is that, first of all, the patients responded very well, even patients that were not responding anymore to the autologous one, although the treatment is more toxic than the autologous one, because these cells are cells that are healthier, and so they are more prone to respond. And in addition, we wanted to study these cells to understand if there was a difference from the cells that were collected from the patients. And indeed, what we found out is that the cells, after they activate and expand in the body of the patients, they have several signs of higher power and higher activity. And so we are now trying to understand how to explore this possibility further and which patients could benefit from this therapy. And with this, I thank you all, and I thank all the people that are involved in this project. Most of all, I need to thank all the families that put their trust in us and allow us to take this journey together and to test this approach. And thank you very much. Yeah.
Can I invite Alan? Do you want to go back? Yeah,
They told me to leave this.
Oh, yeah.
This
Go there. We want them in the middle. I'll go here.
Here? Because they told me to sit here.
Do they? Yeah.
Yeah.
In the middle.
Thank you very much.
Hi.
So, as I said at the beginning, we're introducing Gavin and Alan to join the discussion, which I'll moderate, and I'm keeping an eye out for Donna, who's going to wave right if there's questions online. Right. So, any questions to get started? If you wait for the microphone, thank you. If you could say who you are and who the question's addressed to, that'd be great, thank you.
Yeah, so my name is Lucy Mellon Jameson, so I'm Jude's mum, and we're bereaved parents. And this question is really on behalf of myself, but I think other people in the room now, but also in the future. So as treatments advance, it's great that survival rates improve, but I feel that we get to a situation where there are so many unique cases where you are often told that the data isn't available, you're in the sort of the unknown. And so my question really is, we've got to a point where relapses seem to be so kind of drastic, like my own son's was. We just kept being told, like, we've never seen anything like this before. And what I want to know, I guess kind of for my own comfort, but also if anybody else has the same question now or in the future, is what does their data contribute towards? Like, I know that it will be on there somewhere, but their unique cases, the extent of the disease that they've had, how it had relapsed, the presentation of it was so unlike his original diagnosis. How, sort of like worldwide, how is that being collated and used?
Right, thank you. So thank you for the question, which is really, I think, addressing how we make best use of information on individual patients in order to learn from the disease, because as you say, they're all different. And I guess there's two main aspects of that. You know, there's the clinical side, the sites of disease and the pattern it spread, but also probably, I think your question's getting at the biology, right? What is it we can learn from, say, the sequencing of the tumours and things like that in order to understand what is special and what's driving it. I'll invite everybody else to comment. From my perspective, your question really highlights the importance of the biological studies, which we tag onto clinical trials such as the ones we've been describing. And as Francesca just showed, actually measuring what's happening in the therapeutic cell as we give it before and after treatment, as well as getting the samples from the tumour samples before and after treatment. I think that's how we can really start to learn. And then the second part of your question is really about data sharing. It's making sure that all this data is publicly available. Now, or especially available to other researchers who will be able to interpret it. I think that a lot of progress has been made in terms of this collaboration and data sharing. I don't know if anybody else maybe from a patient's perspective wants to comment.
Well, first off, can folks hear me? Is my mic on? Great. Thank you very much for the question. I greatly appreciate it. I'm a bereaved parent as well. This is my son right here, Evan, who was diagnosed with stage 4 amplified neuroblastoma at the age of 3 in 2006. Then over the course of the next 4 years across 3 institutions in the US, battled multiple relapses, including relapses in the CNS. And treatment failed my son in the fall of 2010. And I'm here with my wife, Wendy, in the back there, and we do this work in honor of Evan. So your question is particularly important to me. I will share 2 thoughts. First, I was told by one of the investigators who was running one of the clinical studies that my son enrolled in, after that treatment failed and my son passed away, that one of the things that they learned about their experience with this agent to treat Evan's disease was that it did not cross the blood-brain barrier, and they learned that specifically from Evan. My initial reaction to that was, well, why didn't we know that before we enrolled in the trial, right? But upon reflection, I appreciate that you can't know everything. As brilliant as our colleagues are, some things are unknown, and our patients teach us, right? Their patients teach them. So I take just a tiny bit of solace in knowing that my son, even though he's not here with us, he's helping the next kid, right, who's not getting that particular agent for CNS-related disease. And then the second point I'll touch on is I've been on this crusade. Sometimes I feel like Don Quixote on this crusade. I think, and the investigators are going to get cross at me when I say this, but I think that every single clinical trial that neuroblastoma, we'll take that as an example given we're here at this meeting. I think investigators for every neuroblastoma trial at the conclusion of the study should send the results to every parent and patient who was involved. Those patients make this work possible. I don't think that is too big a lift. It's an email, unless the patients and parents want to opt out at consent. It may actually be an incentive for patients to enroll in a trial, and it acknowledges the fact that these patients contributed to this work. My wife and I, Evan enrolled in 4 trials, and if we wanted to find out what happened in those studies, we had to go looking for it. And I think it should be the other way around. So hope that's helpful.
Yeah, I think the question really hit home with me because, again, I'm a bereaved parent of Cleena. She was diagnosed just after her 4th birthday and passed away 2 weeks before her 10th birthday after 6 years of more or less consistent treatment. But the question really struck home because someday you'll sit there and unfortunately your child will be a bar on a Kaplan-Meier chart. And if you know enough, you might even be able to pinpoint where they exist on that chart. And like you said, we all benefit from the kids who went before, and the kids that come after will benefit from the experience and knowledge gained as a result of our kids participating, and as a result of us as parents and you guys as parents in the future signing, you know, that consent to participate, which is probably one of the harder things you do as a parent. And the sense I got from your question is, how do we know that piece of contribution to the development for the future patients is actually being capitalized on? So we kind of talk about the why we want that to happen, but I think where John and Francesca can probably help us is the how they're doing that.
Thanks a lot for all these insights. Well, from the scientific point of view, I cannot agree more with John. So we need to study more and more and more, especially with neuroblastoma, which is such an heterogeneous disease, because the more and more we treat patients, the more and more we get to understand which patients can do better with each treatment that we try. And we have seen this already in our small experience, that between the first patients that we treated and the last ones, we partly understood which one could have more benefits, and we are studying more and more from all the information that we gather, which elements can help us understand which can benefit. But this is not enough. We are also focusing on the patients that unfortunately didn't have a good outcome with our treatment, to try to understand why they didn't, and try to improve in the future the tool to tackle the disease also in those cases. But unfortunately, the truth is that all of this takes time, and sometimes the patients don't have time, and this is something that I think all of us are struggling with all the time. We would like to have the results faster, we would like to learn more, but unfortunately we need to do what we can do with what we have, although we are all committed to get the more and more we can from our kids. And it's so true that we need to get back to the patients. I never thought about an email, but I think that this would be a great idea, and we will implement this, because I think we are always very grateful. We all know that we have patients with which we keep in touch, and so with them we share more information, but probably we need to reach out more to the families to let them know what they did, even if the outcome was not what they were expecting, had a true impact.
Terrific, thank you.
Thank you.
Right. So Leona, but I think first here. Thank you.
Hello, I'm Carrie, and I'm mum to Lucy, who you saw earlier on the stage with Gail. Lucy is 15, currently enduring her third relapse. Diagnosed just before her 9th birthday. She's now 15, she'll be 16 this year. So relapse has been part of our lives since 2022, so we're still fighting that third relapse. CAR-T has come onto our radar on several occasions over the last few years, and John, I think we've even spoken in the past. And it's great to see that there is some progress being made and better results. It's certainly a very different story on the parent side, being part of a closed CAR T-cell parent group. We don't seem to see the same successes, but maybe we just talk about the failures. I don't know. So I nearly didn't sit in on this session for that reason, but I'm very glad I did. However, couple of questions, and again, I don't want to sound negative. You know, I'm very impressed by both presentations and the progress that's been made. But when we talk about trying to find less toxic and less harmful treatments for our children, when I look at some of the treatments as part of CAR T-cell, that doesn't, for me as a parent, sit in the same category. So some of the side effects you talked about is chemotherapy. So for a child, that means feeling sick all the time, possibly losing their hair. They're neutropenic, so they can't go to school, can't enjoy day-to-day activities or mix with people. The GD2 causes pain, as we all know, And they're also anemic, so they're probably exhausted all of the time. To me, that doesn't sound like less toxic and less harmful. I also then translate that to the chart that you showed on there, Francesca. So overall, a 63% success rate, but that 63% includes a partial response or a stable response. So that within that 63%, a lot of those children have had those treatments and they're then going to have to go on and have something else because it hasn't quite done the job. So, can you tell me why, as a parent, Lucy's got scans in a fortnight, so we might be looking for some other treatment. Can you tell me why CAR T-cell now, in 2025, should be an option for my daughter? Because as much as I've seen the progress, it still doesn't seem to fit the bill, even in 2025. And I'm sorry if that sounds negative. I can see all the hard work that is going into it, but it just still doesn't seem to quite be there yet.
Thank you for the comment, and greatly appreciated. And I think most pediatric oncologists are optimists, go into the field because of this sort of relentless optimism that eventually we'll discover something of great value. And I think that's where we are really in the field. We recognise that it's been proven to be of benefit, I would say, in leukemia, but at some price and some cost. And it's still being evaluated exactly how good it is. But I think we're only halfway along the journey towards really identifying the best therapeutics. And there's going to be a lot more progress has to be made before we even start to stop that process of acceleration. We're in the intermediate phase at the moment. We're still discovering what the toxicities of the new therapies will be. And, in my talk, I spoke about this armoring, the idea of making these more potent. So, of course, we worry that that may translate into greater toxicity. But the only way we can ever find out is by doing the trials. So we're kind of stuck in this position that there's something which in theory looks like it can really work. In the experimental models we do in our labs, we see that it really works. But we don't really know the exact price there will be until we evaluate it. And obviously that evaluation has to be done within phase 1 studies. So I think the concern about phase 1 studies for CAR T is exactly the same for any other drug or new therapy we're trying. There has to be an ethical approval of a trial. It has to be very well regulated. And the parents and the child, if old enough, have to be an integral part of that discussion. What are the risks? What are the potential benefits? What, is this informed consent? The child is the most important thing in that process, but it's only through the trials that, A, your own child could potentially get a benefit, and B, we will learn for the future. So that's where we are, I think. Francesca, do you want to?
I think that, again, there are so your question is about what is the true significance nowadays of CAR T cells for neuroblastoma. And I think that, again, there are two phases to look at this question, the scientific one and the parents one. From the scientific one, we still have a lot to do. We know that some patients respond. We still have to understand exactly which patients respond, why they respond, which one remain in response. The 63% that I showed you before are complete remission and partial remission. There's no stable disease. Some of them have required additional treatment, but not all of them. And then there is also more complex history because the evaluation of the response in neuroblastoma is very complicated, especially for patients that have a long history of disease. For example, 3 of the patients that were having partial response, they are now 4.8, 4.2, and 5.5 years after treatment, and they still have what we call scientifically stable disease. So the imaging still are showing the same thing, but the disease is frozen. So we did some biopsy and we showed that it's differentiated. So there is still a lot to learn also in analyzing the response. What does it mean? It means that probably in 10, 15 years, we will be able to give maybe part of the answer to your question today. We don't have the answer now. We don't know exactly which patient will benefit. And this comes, of course, the treatment comes at a cost. And I really wanted to show you that it's not an easy treatment, and I agree with you on that. It's again a balance, and it depends on several factors, and every time that we discuss with the patient this option, we really go through all the aspects to really understand whether this could be a benefit for the patient or not. Just to tell you like a couple of stories, CAR T cells, when they were first tested, the very, very first testing was in solid tumors, and it was a complete disaster. And so the research was dropped for a while. Then the researcher decided to revitalize the field, and they tested it in leukemia, and boom, it turned out that it can cure a significant proportion of patients. What does it mean? It means that we need to, again, research and understand what is the potential of all this treatment. And the second story is about the dinutuximab, or the GD2 monoclonal antibody. When they first tested it in patients with full-blown disease, the results were not so great, but nowadays it's giving a true impact on patients when using consolidation in first line. What does it mean? It means that we need to learn a lot, and again, with CAR T cells, this is exactly the point. We still need to learn a lot. Then there is the parent point of view of the question, and that's completely different, because as a parent, I can imagine that this response is not enough for you. And as a parent, from the parent perspective, definitely there are a lot of aspects that we need to evaluate when thinking about CAR T cells. And I really think that each situation is different from the other one, and it really needs to be carefully weighted in the situation of each single patient.
I would just say quickly, thank you for asking such an important and direct question. That's the value of meetings like this, right? And it's an honor to be here with Lucy and all the patients. It's tough as a parent, right? Particularly, I think, a bereaved parent, to always try to find the silver lining, but I do. And I think the silver lining here for me is I would have loved for Evan to have been in a position, and mom and dad to have been in a position to think about enrolling him in a CAR T trial. That wasn't the case back when he was in therapy. There was no options there. So that is a positive, right? There are opportunities here in Europe and in the US, as was so eloquently discussed. I think the challenge for parents, I know this would be a challenge for me if I were sitting in the audience with a patient. What do I do? Where do I, because Wendy and I, like all of you, I'm sure we would have gone anywhere and done anything, right? So the question becomes, where do I go? And what's best? And who do I talk to help get those answers? And I think those are really important questions, because I think you have to talk to a lot of people, my personal opinion. You've got to talk to a lot of people and gather a lot of information and make the decision that's best for you and your family. And that's just an added burden I appreciate, but again, I think it's better to have those opportunities than to not have them at all. So the progress is great, but it's not nearly enough, to your point.
Alan, did you have anything additional as well to add?
I think Gavin's point of, you're thinking about where you should go and what option you should take. There's an option, right? I remember saying to our oncologist that the thing I know, because I work a sideline, not my sideline, it's my actual job, but I work in pharmaceuticals, and it is, yeah. But the thing is, I know now in my heart and soul that in 10, 15 years, Clina's journey would have been completely different, will be completely different. I don't know whether the outcome ultimately will be different or not, but the journey, the toxicity, the side effects, the challenges, the loss, things that she missed out on will be different, because trials are going to improve, medicine's going to improve, and there will be more options and more refined options. So.
Thank you. Leona and then Andy, I think.
Yes, thank you. This discussion is just so powerful. So, Francesca, I wanted to ask about your eligibility criteria that you shared, because back in our time, we were driven by eligibility criteria. What can we look at? What would we be considered for? And I'm thinking about the cutoff point for disease burden. And is there anything that you could share about what percentage, how many children will be of the relapse refractory cohort would actually be eligible because of that low disease burden?
That's a very difficult question to answer, to be really honest, because we've been discussing this intensely. We don't have data on the low disease burden of the cohort of patients treated with the conventional treatments, because this is a criteria that we have included in our study. So we are digging into the data to get this response, because of course this is crucial for the trial. So the true answer is that I don't have this answer. We think that the idea that we are discussing in the steering committee is to propose to the patients to have a reinduction strategy using chemoimmunotherapy. And if you consider the results that have been published with chemoimmunotherapy, approximately 50% of the patients obtain partial response and complete response. And plus, we need to include also the patients that achieve a stable disease, because we include also those patients. And from this calculation, we think that approximately 60% of the patients that have a relapse will be able to be included in the study. But this is really, really an approximation. We need to get more data to respond to this question.
Andy, yeah, thanks.
Yeah, we just... Can you hear me?
Yes.
Yeah, we just didn't want Gavin's comment about 10 minutes ago to go unresponded to. So, I mean, I think we absolutely support your idea. We think it's a very good idea, you know, when the study is published, for a lay summary to be sent to the participants. So it's an excellent idea. I think you'll get frustrated because of the time lag with that. But I mean, that's something we're all trying to you know, to improve on. But just, you know, I think definitely this table is supportive of that.
Thank you very much, Andy.
Right. Okay, 3, excellent. I think this gentleman in the stripe was first.
Hi, my name's Aaron, so I'm Jude's dad. Lucy spoke about him a little while ago. I guess I've got a thank you and a question. So a thank yous for Professor Anderson and Dr. Del Bufalo. John, you answered an email late one evening asking for help for Jude, and you pointed us onto the trial in Rome, and we fetched Jude in May 2023. And unfortunately, his disease had progressed at that time, so we couldn't continue. But nevertheless, thank you for your help and the hard work that has been and is continuing. I guess in terms of a question, Jude relapsed with leptomeningeal disease in November 2022. And my understanding is whatever the malignancy, it is difficult to treat and it is generally devastating. So in terms of the current generation or the next generation of CAR, would that go some way to meeting this unmet need?
I'm tempted to say Francesca's best place to answer that, but I think, I'm optimistic about that. You know, the Stanford group were really the first group who started giving direct injection of anti-GD2 CAR into the cerebrospinal fluid, the fluid which bathes the brain. And, you know, they've reported patients with metastatic deposits in the spine having had regressions, in fact, complete remission and sustained remission in one patient following that administration. So it shows that, you know, the CAR T cells, if given locally, can treat leptomeningeal disease. Very few patients, not enough for us to comment conclusively, and it's quite new. But also some patients who get intravenous administration are showing brain responses as well. And Francesca has more direct experience of that than I do, probably.
Yeah. So first of all, we have seen in a few patients that the CAR T, neuroblastoma patients I'm talking now, that the CAR T infused intravenously can traffic into the CSF, as John was saying. So potentially they could. And we have the trial that we have now activated for brain tumors. So it's diseases that are different than neuroblastoma. We give the CAR T cells intravenously, and they can go into the tumor in the brain. So theoretic and the experience from our American colleagues are showing promising results. So there is the potential there, and we are seeing some responses. But I would say that first of all, I'm not sure that we can compare brain metastasis of neuroblastoma with brain tumors, because it's different biological diseases. We have had 4 patients that we treated with CAR T cells that had before CAR T cells brain metastasis, but we don't treat patients yet with active disease in the CNS because we don't know the true potential of CAR T cells in the brain, and we feel that it would be unethical to give CAR T cells for treating the disease. So we still need to get to know exactly what is the potential in the brain for neuroblastoma. For brain tumors, we are seeing some responses. The toxicity is completely different. We have CRS, but we have more toxicities that are related to the site of the disease. And so the treatment of the toxicity is also more aggressive. And so we still need to figure out a lot of aspects to understand how they can work in the brain field, to be honest. And also, in the field in general, we don't know whether intravenous infusion or intrathecal infusion is better, or whether you need lymphodepletion or you don't need. It's a field that is still very, very early, much earlier than neuroblastoma.
If I could just briefly respond to your comment about ethical concerns, and I completely take it for what it's worth, and first do no harm, right? I think we all get that. As the father of a son who had, again, CNS disease, and seeing the paucity of treatments for CNS relapsed neuroblastoma, not only when my son was in treatment, but here we are 15 years later, and we're still in this same boat of really, really struggling for these kids, and we know what the outcome is for those kids. I think from a parent perspective, from this parent's perspective, there is an ethical question to not exploring what CAR T cells can do for children with CNS relapse disease. So it's much more easier for me to say that than you. I appreciate that. But as I'm staring at my son looking at me here in the screen, I would be remiss if I didn't make that point. I think parents would be willing to accept that risk benefit, knowing what's going to happen with no treatment.
I think it's really important, and something Zoom changing is involving the parent in what the question you're trying to answer is. I think that's becoming more and more, one, important, and two, permissible. I think so. That's.
Thank you. That's about
Thank you.
over here, please, I think, and then yellow. Nothing from Donna, right? Not yet.
Hi.
Oh, yes, there is. Okay, so you've got online as well.
Hi, I'm Mandy. You heard from me earlier. I'm mum to Peter. I'm actually sort of following on a bit from Carrie's question about how parents go about making decisions. And this treatment obviously involves an element of chemotherapy. And this might seem like a completely daft question, so forgive me if it's completely unscientific. But if your child has previously shown little response to chemotherapy, which my son did through frontline treatment, rapid Kojak, barely changed his tumour at all, and it was actually the skill of the surgeons that got rid of the tumour. And then on the Beacon trial, when he was on chemotherapy only, he progressed. Why would a parent whose child has not shown any response to chemotherapy previously be comfortable accepting a treatment that involves an element of chemotherapy? I don't know whether it's different in this particular treatment, but it would be interesting to know whether that would immediately say, no, you shouldn't do this, because actually through my son's relapse treatment, they decided not to do stem cell transplant because there was no point putting him through the burden of that high toxicity for what might be no benefit for him whatsoever. Is it the same for CAR T therapy, or is it different?
Not a stupid question at all. Excellent question, thank you. And quite a simple answer, which is that the benefit of a chemotherapy for preconditioning for CAR T cells is not dependent on any sensitivity of a tumour. It's given simply to decrease the number of competing white blood cells in the blood. So even patients with chemo-resistant tumours will get the same beneficial of the chemotherapy given prior to CAR T cells. And it's a smallish dose, just given once, in fact. Right, shall we go to Donna then? Oh no, sorry, apologies, yes.
Is it working? Yeah. Oh, hi, I'm Kelly. My daughter's currently fighting her third relapse. We actually came out to Rome to start CAR T, but unfortunately her cells didn't express enough GD2, But we're so thankful to Dr. Francesca for actually testing her tumour to see if it was expressing, because otherwise we would have put her through that treatment and it would never have worked for her because the T cells wouldn't have interacted with her neuroblastoma cells. So I guess my question is sort of for you, John, because would you be testing tumours in the UK to see if they have GD2 expression before those children go for CAR-T?
In our previous trial, we didn't, because we didn't have a good enough technology to do it. We've now introduced it, but more importantly, we're testing for the other, target antigens, and it'll be written into future trials, I think.
Okay, amazing. Thank you.
Right, Donna.
Thanks. Kelly, that was the first question I had. Second one is there's a lot of CAR-T activity going on in China, and we have an online dad from China. Can you comment at all on some of the advances there? Are you aware of any particular outcomes from?
I am aware, I follow it as closely as I can, but many of you will be aware that the results are always published as rapidly as we see. So some of the results are in abstract form. So although I keep an eye on it, I don't have a strong sense of large cohorts of patients who've had their results published in a detailed way. But that might be, my ignorance more than that. Anything else?
No, me neither. I have the same. I know about other diseases, but they have published in other fields. But to the best of my knowledge, not in neuroblastoma.
Was there anything else, Donna? Okay, more questions. Yes, leave.
Thank you so much. So the first one is like a remark. When we were in Saipan in Greece, the parent organization asked us if we could also do the publications as open access, because then they didn't need to pay for the papers to download and to see them. And I think this was a really, really good suggestion. If I went back to my old publications, I still need to pay for them because I even cannot access them myself. So this is a funny thing. So I think we really are aiming, I hope all of us, to make the publications open access. So even if you cannot email this to the parents themselves, there might be a way, for example, to let the people know that the paper is out there and where we can access them and that people don't need to pay. Right. So that was really my first remark, that we understand we need to feedback, and we are trying to do this step by step. And my second is like a more general question. So we will speak about the liquid biopsies and those novel technologies later on in this symposium. And I think we might also need to sit together and to see how can we also implement, for example, the novel technologies, or maybe not so novel technologies, in also the CAR-T trials. Maybe these will already help us to identify the responders and non-responders, or so this is something not a really question, but just a plea. And my third question is, there are different options coming on. So there will be different CAR-Ts, there will be 7H3, GD2, maybe other also novel. How on earth are we going to identify which patient will need what? I don't know if there's no simple answer there, but I think that will be the next question, which I think we already also touched upon this morning. How will we decide, we as clinicians and you as parents, what we want to do next?
I'll just say a few comments about that, Lee. Thank you. I strongly agree of the importance of coordination in this. I mean, one of the concerns is that with so many, all these trials are very, very complex and very expensive and require a lot of funding and expertise and specialist centres for manufacturing at this stage. Hopefully in the future that will all change and be democratized. That's the reality of how it is at the moment. And coordination is essential to prevent people asking the same questions in different clinical trials. And we are, well, many of us are actually really very actively working towards that coordination at present. Yeah. Did you? Cormac.
Thank you very much. Francesca, you highlighted the problem of the children with not low burden disease at the time of CAR-T. The question is, maybe John, all of those trials that you listed, you think that that's going to be a problem for all of those other CAR-T cell trials? Is that built into the eligibility? How are we going to address that challenge?
Yeah, it's a great question. It's kind of a philosophical question, isn't it? I mean, it's true that both in the patient experience and in animal models, the small burden disease is more curable. But I firmly believe that it makes no sense to say that large tumours can't be treated by this eventually, and will be. So I think it'll be a shift, it'll be a moving parts thing with a shift in our emphasis and philosophy, but it kind of makes sense at the moment to try and give the most patient benefit we can by targeting smaller tumours until we identify the engineering and combination treatment effects, which will really start to show the biggest benefits and shrinkages for the larger tumours. So I'm optimistic. Yeah, I mean, I really believe that the large disease burden will be. You've had some patients in Rome with big disease burden with spectacular results. It's just there's a much higher statistical advantage in the small disease burden. But your experience proves it can be done.
Then we also need to understand, I think, why the higher disease burden is impairing the activity of CAR-T, because it probably is related to the tumor microenvironment, so we are studying this. And so it could be well the case that, for example, Claudia's treatment, since IL-18 has the ability to shape the tumor microenvironment, maybe in her case this will not be a challenge. So I'm optimistic as well. The more we learn, the more I think we can improve.
Gudrun, I'm going to come to you in a minute, but you're second in line.
Me again, sorry. This is more of a general question, given that it's a general question about clinical trials. So given that we are all part of CIAPN here, so we've got Italy and the UK, do you think there will ever come a time where parents like myself, and I know there's other families in this room, will not have the burden and the cost of having to travel for a clinical trial? Do you ever think that a time will come where a child might be eligible for your trial, Francesca, in Rome, but actually if you coordinate with John, a huge amount of it can actually be done in the UK? Now, I'm not new to clinical trials. Lucy's been through 3 UK clinical trials, and we've also travelled to Dr. Scholler in the States twice. So we're not new to clinical trials. We know how they work. We know the data's essential, all that kind of stuff. But for us, as you can probably imagine as parents, not only have you got the burden of my child needs this treatment and my child is sick, but now I need to put him or her on a plane to Rome or on a plane to MSK. You know, do you envisage ever a time where, you know, now in the day of technology, in the modern day that we live in the age of technology, where parents like myself might feel less burdened by travel?
Yeah. I'll let Alan and Francesca address.
Can I maybe just add to that slightly different parent perspective on that as well? Like, obviously, we would all love to avoid the burden, but sometimes what we also need to think about is there's expertise that builds because of execution multiple times of the same type of activity, right? And I would rather, burden aside, if I was to be able to have the financial and travel burden taken away, I would rather have my child go to a centre for a specific type of treatment where those people are routinely delivering that treatment and have the expertise. That it's not because, while it doesn't feel rare in this room, neuroblastoma is a rare disease, and the opportunity for doctors and oncologists to actually practice those approaches are small. So sometimes we need to balance that. There's a burden of travel, 100%, but there's also the advantage of expertise and numbers. And that may never, for a disease like neuroblastoma, those two things may never even each other out.
I appreciate that, but you've got two centres of excellence here delivering very similar, don't think this microphone's working, delivering very similar treatments. And also from my own experience, so as an example, Juliet's here, we were part of the minivan trial. So we got on a plane for 33 weeks, every fortnight, taking Lucy to Southampton for nivolumab, which she could quite easily have had at our home hospital because they are experienced enough to deliver it. I'm not saying the whole thing, but that, could there be parts of the trial that allow you to do it at your home hospital because it doesn't require that extreme level of expertise. We also did naxitamab at Leeds. I wouldn't have dreamed of trying to do that anywhere else because it's a horrific treatment. It requires very specialised nursing and care, and so I was happy to travel the hour and a half every other day to Leeds for that. So I totally agree with you, but surely there's got to be some commonality in some areas that say, listen, this is the treatment you require, but you can have this in your home hospital because it's
even
reasonably easy to deliver. So
thank you for. I'm going to invite Andy Pearson to respond, and Francesca may respond with some detail about our plans as well.
So, very clear answer. I personally believe absolutely the answer is yes, and people are trying enormously hard to do it. I cannot promise you it'll happen in 2026, but definitely the clear desire is there to do it, and people are trying enormously hard to achieve that. So I think that's the answer is yes, definitely. And all the arguments make enormous sense. So it's just the practicalities, and people are trying honestly hard to do it.
I agree with Andy. The direction is that. I think that we will get there. It also depends. I think there is probably not a single answer for all the trial because trials may be different. So it may depend. We are aiming at opening as many centers as we can. I showed you the list of the ones that were the first to participate, and so they are the more involved. But the more we receive requests, the more we would like to include the centers. So that's the future. But there is a lot of technical challenges to that. It's not that easy.
I would just add maybe another layer of complexity, because I'm good at that. I think about the people who aren't in this room, right? And maybe they're online, but just think if you're in Africa or in another developing country, medically underserved community, population, and you're dealing with this nightmare. I hear you loud and clear about your challenges, and you're in one of the greatest countries in the world, right?
Thank you.
So, sometimes
way to win the home audience.
Yeah, sometimes I wish I could stay here longer. But I think about those people, right? And it's such a complex set of challenges, and I don't begin to have any solutions. But I think as we think about things like CAR T-cell therapy and how expensive it is and the high-tech equipment required, on and on and on, hopefully we get to a place where the economy of scale kicks in and these things become less expensive and more accessible to more people, not only in the US and the UK, but around the world.
Fantastic. Donna? Oh, Donna, yeah. So we have Gudrun Schleiermacher and Donna, and that has to be the end or be over time.
Donna?
I thought Gudrun was going to go first.
Yes.
Go ahead, Gudrun.
Yeah, thank you. So I'm Gudrun Schleiermacher from Curie Institute in Paris. And I wanted to ask you, Francesca and John, it's already been said, but maybe to restate what your vision is with regards to CAR T in the overall strategy of treatment for relapse. Maybe that will be addressed later today also. But we heard this morning that treatment for neuroblastoma at diagnosis is in fact composed of different phases, different components, and the different blocks, how they're organized together is crucially important. How do you think the CAR T treatment you have so nicely explained will find its place in the overall treatment strategy? What is your vision here?
Thank you. Do you want to start?
Okay. There's two parallel pathways. One is improving the technology in an incremental way with a series of coordinated phase 1 studies. And the second is to try and bring what is currently the best thing into upfront treatment within a randomized fashion. That would be my vision.
Yeah. Yeah. So if I may share, my true dream would be and it's what we've been discussing, to see whether CAR T-cell in the future could ever replace the high dose treatment, because we all know the burden of toxicity, long-term toxicity that are associated with high dose chemotherapy. So the ideal setting would be to compare in a randomized fashion high dose chemotherapy with CAR T-cells, and if they compare at least favorably, similarly, to substitute the high-dose chemotherapy with CAR T-cells. That's really my dream.
Right. I think it is 12:30 exactly, so we're spot on time. And yeah, I'd like to thank the panelists. I'd like to thank Francesca for flying over this morning and flying straight back today and parting from her family on a Saturday. And thank everybody for their amazing contributions and an excellent discussion. Thank you for coming.